Okay. This is my PowerPoint for chapter 4. So, we're mostly going to be talking about bacteria cells. We're going to be talking a lot about the external boundary and internal features of the bacterial cells. And here's a picture here with the appendages and things we'll be talking about. So before we get started, we'll go over some basic understanding of bacteria. So here is a list of things that all bacteria has. So all bacteria has a cell membrane, cytoplasm, ribosomes, and chromosomes. Some but not all bacteria have cell walls, phalangeal, membrane, S-layer, cytoskeleton, inclusions, membranes, intercellular, nanowires, endospores, microcompartments, and glossocyra. So this is essentially what all bacteria has, but this is some, but not all bacteria has this little subcategory of things. And in the next slide, you can kind of see, I put this picture of this bacteria cell in here just because it has everything kind of listed that I just talked about. And now it varies, like some of these things vary, but like I said, with the cell wall would be something that's not in it. However, say ribosomes would be, and cytoplasm. So I'm going to talk a lot about shapes and arrangements of bacterial cells. So the average bacteria is about 1 micron. So that's just fun fact. So there's 3 main shapes to a bacteria cell. The first one is the cocci. So the cocci bacterial cells is spherical shaped or ball shaped. Some can be more oval or bean shaped. Even some can be pointy. And the next main shape I'll talk about is the rods. So the rod bacterial cells can be blocky, spindle shaped, round ended, long, and thread like, drumstick shaped. And lastly, the last bacterial cell shape I'll talk about is the curve shape. And that's more of a spindle shaped cylinder, brigid helix, corkscrew shape. And so I put this picture in here, and this kind of shows you more about what we were talking about in the last slide. So this is the cocci. As you can tell, bacteria can be very varied in many different types of subcategories. So like cocci, obviously, like this is spherical ball shaped, but they're kind of separated. However, this one is more of a chain together. So they all have different variations to them, but those are the 3 main shapes. And here's the names of the more of the subcategory per shape that you have here in this diagram. So we'll move on from that. But back to shapes and arrangements. So the main factors influencing the arrangements of a particular cell type are its patterns of division and how the cells remain attached afterwards. So cocci cell types are the most diverse in variation. So when it comes to arrangements, they have the most diverse variations that could be. So like they got single arrangements, pair arrangements, group of 4 arrangements, irregular cluster chains of a few hundred, qubit packet of 816 or even more cells. And on the left here, you can look at the names. They're too hard to pronounce. I think, but I know Coxy but yeah, you can keep up with those. So I'm going to touch base on a biofilm and what a biofilm is. So essentially, a biofilm is a bacterial habitat. Most bacteria lives in cooperative communities where bacteria really can thrive. Uh, with many other bacterial species like other bacteria, archaea, fungi, and algae. So bacteria really gets along with those other things. A biofilm has anaerobic microbes near the surface of the oxygen, uh, where the oxygen is high, and they have anaerobic microbes near the bottom where the oxygen is low. So that's kind of how uh, a bacteria habitat is, a biofilm. And this picture here is a good example of what one would look like. It's out of the textbook. I liked it. So talking more about motility, the flagell or little propellers, that's what helps the cell move, or the bacteria. So the primary function of flagell is to provide motility by self-propulsion. And in the top right-hand corner here, this is a picture of what one looks like. And there's three major parts of this, and this is the filament, the hook, and the basal body. And you can see where those are labeled. And the filament is a helix structure composed of proteins. So talking more about bacterial movement, bacteria can move freely, can move very freely, and can even respond to chemical signals called chemotaxis. They rotate counterclockwise in a smooth linear line towards a stimulus. That action is called a run, and runs are interpreted as tymbals. And this picture here kind of shows, kind of how they move. Very angular, I guess. Very runny, like self-proportion, like so. So that, I hope that goes over the motility part. So I'm going to talk about some polar arrangements of a cell. So the phalanges are attached to one or both ends of a cell, and there's three main types of that, and that's a single phalange, and you can see in picture A. how it kind of just has just like a single tail, and it's single. And you got number C here, small bunches and tuftles of phalangia emerging from the same site. And the C right here, you can see where it's all bunched up. And with the D here, with phalangeals at both poles of the cell. And I can't really tell by that picture, but it's out of the textbook, so I'm going to go with it. So, talking more about internal structures. So, cytoplasm, so the contents of cytoplasm and what sort of cytoplasm is. So, cytoplasm is what makes up most of your internal cell. And the major component of cytoplasm is water. About 70% of it is water, and it acts as a good solvent for the cell. And the other 30% would be a mixture of sugars, amino acids, salt, chronomate body, ribosomes, gnarls, and fibers would make up the other 30%. So that's a part of the main internals. And remember, all bacterial cells have cytoplasm. And to move on to ribosomes. Ribosomes are also an internal structure. They're made up of RNA and proteins. About 60% of ribosomes are RNA, and 40% of ribosomes are proteins. Ribosomes are responsible for protein synthesis, and they are also scattered throughout the cytoplasm. And here's a picture of a ribosomes with the large subunit and the small subunit. So, bacteria, chromosomes, and plasmids. So, bacteria do not have a nucleus because its DNA is not enclosed in a nuclear membrane. Instead, DNA is in a dense area called a nucleotide. Bacteria DNA is a double helix formed in a circle. So its DNA is more in a circle form, and the DNA divides in genes, and that promotes cell growth and maintenance and things of that nature. And this is kind of what it looks like. So we're going to talk about the cell envelope, and it's pretty much comprised of 3 basic layers here, which mostly 2, but it's mostly the cell wall and cytoplasm membrane, and then you got like kind of other, more of the membrane too. So the boundary part of bacteria, the cell wall, is important to define the shape and to add structural support to the bacterial cell. The cell wall is made up of peptide glycerin. Peptide glycerin is made up of sugar chains that's connected to the short protein fragments that provide the framework of the cell. These chains are called glycine. And I added that picture from the textbook, and this shows like the glycine chains and how the cell is made up. And I just thought that was kind of neat. And lastly, the last thing that we're going to talk about is cytoplasmic membrane. So cytoplasm membrane appears just under the cell wall. Cell membrane is a lipid base with proteins embedded to varying degrees. The structure is primarily phosphate lipids, which makes about 30 to 40% of its total mass, while 70 to 60% of its mass comes from proteins. The main function of cytoplasm membrane is to regulate the transport in and out of the cell, and the membrane is also selectively permeable, so that means it has control of what comes in and out of the cell. Kind of with the cell wall, kind of runs together. And with that being said, that is the end of my PowerPoint. Thank you.